Assalamualaikum. Now we are here with the FSC Biology Chapter 1, best line summarized for PPSC lecturer and subject specialist biology and zoology. Respiration. Bacteria can be aerobic, anaerobic, facultative or microaerophilic. Aerobic need oxygen, anaerobic don't need oxygen, facultative don't need both, whether present or not, or microaerophilic need a less amount of oxygen. Facultative bacteria can grow in presence or absence of oxygen, and they do not care whether the oxygen is present or not. Microaerophilic bacteria require very low amount of oxygen. Pseudomonas is aerobic bacteria, spirochete is anaerobic bacteria, E. coli is facultative bacteria, and Campylobacter is microaerophilic bacteria. Please remember all these 4 examples because these are very important in regard of exams. Growth and reproduction. Bacteria reproduce asexually by binary fission, and meiosis is totally absent in bacteria. During cytokinesis, cell wall grows inward to separate. Cytokinesis means division of cytoplasm. What is generation time? Generation time is interval until the completion of next division. So from one division to the next division, the time interval is called as generation time. Now these 4 phases of the growth curve are important to you for understand. Lag phase, preparation for division. It is the phase for preparation of division. No growth occur in this phase. Log phase, rapid growth at exponential rate. Exponential rate means at high rate. Stationary phase, death rate equals reproduction, means the death rate and reproduction rate become equal and the population of bacteria remain same. Decline phase, start dying. The bacteria start dying, death rate is more than reproduction. In this phase, the population of bacteria start dying or decrease. So these 4 phases of growth curve are important to you to remember. During conjugation, donor bacterium transfer genetic material to recipient. They use sex pellet to transfer material. Conjugation produces genetic combination for better survival. Importance and physical control. Bacteria recycle organic matter and cycles of nitrogen, phosphorus, sulfur and carbon depend on bacteria. So these cycles will not work if bacteria is absent. They can spoil the food and vegetable. This is common importance. Approximately 200 species are known to cause disease in human. Means 200 species which cause diseases in human. Now, what is sterilization? Sterilization is destruction of all life forms. Which life forms? Life forms which are present on the instrument or utensils which we need to clean or present in the lab. So the destruction of all life forms present in the instrument or utensil we need to clean is the result of sterilization. This is basic sterilization. Physical factors like steam, dry heat, gas filtration and radiation are used in microbiological labs. Remember that in microbiological labs, high temperature is used to control microbes. Moist heat causes coagulation of protein, while dry heat oxidize chemicals. This is the reason behind using moist heat and dry heat. Electromagnetic radiation below 300 nanometer are effective killers like gamma rays. Heat sensitive, we can say that cannot be sterilized by heat, so they are sterilized by filtration like antibiotics, serums, hormones, etc. Chemical control. Antiseptics inhibit growth of living tissue and disinfectants are used for non-living things. Please remember the difference between them. They are oxidizing or reducing agents. Disinfectants are oxidizing or reducing agents like halogen, phenol, hydrogen peroxide, potassium permanganate, alcohol, and formaldehyde. Alcohol and formaldehyde are most common disinfectants. Antibiotics and chemotherapeutic agents work with natural immunity and stop growth of microbes. They work in living tissue. Antibiotics work in living tissues. Examples are sulfonamide, tetracycline, penicillin. Microbicidal kills microbes immediately. They may be drugs or antibiotics. Microbistatic inhibit reproduction. They only inhibit reproduction and keep constant the reproduction of microbes. Immunization and vaccination. In 1880s, Pasteur isolated bacterium responsible for chicken cholera. He used the technique devised by Robert Koch. In his public demonstration, chicken failed to get cholera due to old culture. What happened in the public demonstration? Actually, Pasteur was saying that these organisms were responsible for this disease and he injected these organisms in the chicken in front of the public and wait for the chicken to get disease, and the chicken didn't get the disease because the culture he used to inject was very old, and that's very famous and common story. He found bacteria lose their virulence when become old and chicken become immune. That's why he did not infect. So that was another discovery based on that mistake or demonstration. He used attenuated or less virulent culture to treat anthrax and was successful. Pasteur also made a vaccine for hydrophobia or rabies. Edward Jenner injected cowpox material to James Phipps. The name I want to revise James Phipps, which was the boy who was first injected cowpox material and then he became immune to smallpox disease. Antibiotic. Antibiotic is a Greek word meaning against life. Against life means against microorganisms. Antibiotics are secreted by bacteria, actinomycetes, and fungi. These are three sources of antibiotics. Antibiotics synthesized in laboratory also originate from living things. Misused antibiotics interact with metabolism and may cause death. So avoid using antibiotics on your own. Misuse of penicillin cause allergic reactions. Misuse of streptomycin cause deafness. Misuse of tetracycline cause discoloration of teeth in children, also in youngs. Cyanobacteria. Cyanobacteria or blue-green algae is largest photosynthetic group of bacteria. They are also prokaryotes. They are true prokaryotes. Trichomes are chain of cells, and these chain of cells form filaments which are surrounded by mucilaginous sheath. They are gram-negative cells. Cyanobacteria are gram-negative cells, lack flagella, and move by gas vesicles in water. Please remember that they move by gas vesicles. Like eukaryotes, they have photosystem too. Cyanobacteria used phycobilin, which is a group of pigment, as accessory pigment located in thylakoid. Phycocyanin, which is also phycobilin, it is a blue pigment. That's why they are called as blue-green algae. Reserve food is glycogen, not starch. Harmogonia, aconites, and heterocysts are present in cyanobacteria. Heterocysts help in fixation of atmospheric nitrogen. Please remember this MCQ because this MCQ has already been asked in the PPSC exams many times, that what is the function of heterocyst. It helps in fixation of atmospheric nitrogen. Oscillatoria is a pollution indicator cyanobacterium. What is pollution indicator cyanobacterium? Which is pollution indicator cyanobacterium? Oscillatoria. They form association with angiosperms. Many species form water blooms having unpleasant smells. Water blooms are famous for their unpleasant smell. Super blue green algae is expensive pond scum, which is a photosynthetic bacterium, and its food has 60% protein. So super blue green algae can be used as protein source. Nostoc. Terrestrial nostoc is terrestrial subaerial cyanobacterium distributed in alkaline soils. Unbranched trichomes appear beaded like beads. Large, round, light yellowish, thick-walled cells in filament are heterocyst. Please remember that this is the definition of heterocyst. Harmogonia are fragments which are formed when filament breaks near heterocyst. Harmogonia are reproductive fragments. Actinids are thick-walled, enlarged vegetative cells which store food. Why they store food? Because during unfavorable condition they have to store food and they become inactive and wait for next favorable condition and become resting cells and convert into normal cells during favorable condition. Please remember this. That's it for today. Next chapter, inshallah, will chapter 7 complete concept and summary. Wait for the next video. Now I shall check whose comment will depend on this video. Allah Hafiz.